lecture, and Professor P. Balram will be chairing the session.
Good morning. It's a special privilege for me to introduce the EMBO keynote lecturer this morning, Professor Chris Dobson. Professor Dobson is a fellow of the European Molecular Biology Organization. And to tell you a little bit about Professor Dobson, very briefly, he did all his education right up to his PhD at Oxford University. Subsequently was an assistant professor at Harvard University for 3 years before coming back to Oxford in 1980 to join the faculty. He remained on the faculty at Oxford right up to 2001, when he moved to Cambridge as the John Humphrey Plummer Professor of Chemical and Structural Biology. This, I think, in Britain would probably be the equivalent of a leading Labour politician defecting to the Conservative Party. He's currently the Master of St. John's College, and he's been very widely recognized for his work. Fellow of the Royal Society, Fellow of the US National Academy of Sciences. He's been the National Lecturer of the American Biophysical Society, and also the President of the Protein Society. He's delivered the Royal Society Beckerian Lecture. He's been received the Davy Medal, and from the Protein Society he's been honored with both the Stein and Moore Award and also with the Hans Neurath Award. He more recently received the Royal Medal of the Royal Society, and he's also an honorary fellow of the Indian Biophysical Society, where he has delivered the G.N. Ramachandran Lecture. So it's a special privilege for me to invite Professor Dobson to give his keynote. Professor Dobson.
Well, it's absolutely wonderful to be, you can all hear me? It's absolutely wonderful to be back in India to see so many, I don't call them old friends, but friends of long standing, and also new friends here, many friends in the audience. I should start off by acknowledging the fact that my visit here has been sponsored by EMBO, and as you may know, it's a major life science organization in Europe. It funds many different things, it publishes a number of journals, and in particular it gives fellowships to people for short or long periods of time, and I think many people who work with me have either had such fellowships or received fellowships to visit elsewhere. This wasn't written by me, but they think they run the top scientific meetings in Europe, so I've just repeated that on my slide. They're very good. So what I want to do is to talk about the so-called amyloid state of proteins, a phenomenon that really we stumbled into about 25 years ago, and it's increasingly recognized as being an absolutely key factor in many of the most debilitating diseases of the modern era, and I'm going to mention that as well. It's also very strongly coupled with intrinsically disordered proteins, and I'll try and bring that element out as well. And I want to try and demonstrate the breadth of biophysical and biochemical and chemical techniques that we've tried to bring together to solve some of these issues and go right the way through to potential therapies. So, as I'm sure you're all aware, proteins are extremely important molecules, and the correct assembly of proteins and other components within the very crowded cell is absolutely critical for life. In folding, I include intrinsically disordered proteins, which are folded in a different way, but actually it's getting them in the correct structure is just as important as globular proteins, because they have to exist within this complex environment and form the right complexes at the right time to give the right function. And these are all vital elements in establishing the homeostatic process of proteins within cellular systems. And because protein folding or protein structure is so important, if you fail to maintain the correct fold or the correct structure of the protein, it gives rise to many different diseases. And they're now known to be really three types of disease associated with misfolding. One is when the misfolding process is picked up within the cell, generally the endoplasmic reticulum, and degraded by quality control systems. And cystic fibrosis is the classic example of a protein misfolding disease where you don't have enough of the correct protein, normally because of mutations in the gene. Some other proteins misfold and end up in the wrong place in the cell or the organism. Alpha-1 antitrypsin deficiency is an example of that. But other misfolded proteins precipitate and form intractable deposits. And these are the ones I'm going to talk about. And these disorders are extremely important. We'll hear much more about them later in this meeting, and I hope that, thank you. I hope that this serves as some sort of introduction, but misfolding can result in the aggregation of otherwise normally soluble proteins to give these intractable amyloid deposits. And there are about 50 diseases now associated with this phenomenon. They include Alzheimer's disease, the prion diseases, type 2 diabetes, Parkinson's disease, Huntington's disease, motor neuron disease, and so on. And these are extremely important. There are about 40 million people suffering from Alzheimer's disease. There are 400 million suffering from type 2 diabetes. And all of these diseases are essentially untreatable, certainly not curable, and very difficult to handle at the moment. And I estimate that approaching 5% of the population of the world has one of these diseases, and that number will increase by a factor of 2 or 3 in the next generation. What's interesting structurally is that they're associated with different types of protein. Some are full-length folded proteins like transthyretin. Others are fragments of precursor proteins like the fragments of the A-beta protein. And many of these are intrinsically disordered, and we'll hear a lot about those today. I thought I'd just give you something of the scope of these diseases, just as an introduction. The most famous, of course, is Alzheimer's disease, and it's the most common form of dementia, and the deposits of these amyloid structures, either as the famous plaques of A-beta, or inside cells, you get tangles also of the protein tau, and this leads to the death of neurons and the many symptoms that I'm sure you're aware of in terms of loss of memory and cognitive ability. But these deposits can be very different in different diseases. For example, if you put people on dialysis for kidney failure for long periods of time, they'll get deposits of amyloid, but in this case in skeletal tissue and in joints. It causes a great deal of pain. It's now recognized that a significant proportion of elderly people who die of heart failure actually are affected by the fact that amyloid can be deposited within the heart muscle. It reduces its rigidity and therefore contributes to heart failure. And it's been estimated that maybe 25% of people, elderly people with heart disease, are actually suffering from conditions associated with amyloid. We actually got into this whole area through a systemic disease where you can get very large quantities of protein deposited in different organs, particularly the liver, spleen, and kidney. Kilogram quantities, huge quantities that give rise to, again, that's a fatal disease like nearly all of these. But we were very lucky because we were studying the protein lysozyme, looking at its folding, and it turned out that although it didn't seem the most interesting biological protein, perhaps by 1995, it turned out that there were a few people who had mutations in the gene encoding lysozyme who ended up with these massive deposits. And this was work with Mark Peppers that we did at the end of the 1990s. And I just wanted to give one example of that. It's all published, although we're still working in this area, because we had derived a sort of folding surface for lysozymes, starting from the largely denatured state And we realized there were different pathways of folding, some more efficient than others, and there were a number of intermediates which were populated on the folding pathway. And the major intermediate turned out to be quite disordered, as you can see here from a simulation based on experimental data that we carried out. But in fact, in that case, the structure is largely preserved within this so-called alpha domain, but this part of the protein is largely disordered. And the fact is that the mutations are all in this part of the protein. And so what happens is the native state is destabilized by the mutations, but the intermediate is not destabilized because the mutations are not in the region which is structured. And so this partially unfolded intermediate can be populated under physiological conditions. And because it's partially unfolded, then it has a tendency to aggregate. And we've studied this in great detail. So we could see here the link between, if you like, normal folding and misfolding and the aggregation of partly unfolded proteins. The other aspect of these conditions, which is very interesting, is that when you extract from patients the deposits that are formed in different tissue, whether it's from Alzheimer's disease or from the lysozyme amyloid disease, you see on the electron micrographs very, very similar structures, which are typically microns in length, a few nanometers across, and with high elements of beta structure, and bind to all sorts of dyes, which you'll hear much more about later, I'm sure. And when we saw these structures, we were immediately sort of fascinated because we knew about globular proteins that all had their unique folds. And then we're seeing this other type of structure in disease, which had really rather a common type of structure, this thread-like structure, whichever disease was involved. And so the first topic I want to talk about is what we could learn about the molecular nature of the amyloid state. And again, I'm sure you'll hear more about this. Like everything that we do, and research councils hate to know it's all done by accident, we were looking at the folding of this small protein. It's a domain, an SH3 domain from PI3, a kinase, and this is one of the most common motifs in the animal kingdom. And we took it to low pH where the protein unfolded. And we did a long NMR experiment to try to characterize its unfolded structure. But we found at the end of the experiment that the sample had turned into a gel, and we got no signal from it. But we knew that the mutational variants of lysozyme associated with disease also formed gels. And so we looked in the sample tube, and to our amazement, it was full of these fibrils that we'd previously thought were only associated with disease. And they had all the characteristics of these amyloid structures that you see in disease. And so we went on to look at other proteins. This is one example. This is myoglobin, the first structure determined of any protein. It's a helical structure. You might think it's the least likely protein to convert into amyloid fibrils. It's very soluble, it's very helical. But in fact, if you take it to destabilizing, denaturing conditions, some of it will convert into these fibrils, again associated with disease. And we spent a lot of time looking at these ordinary proteins in the laboratory to watch how these fibrils formed. Of course, some people said, well, there's clearly a code for the amyloid structure, but we're just not clever enough to see it. And so we looked through a very determined graduate student called Marcus Fandrich at homopolymers, where every amino acid is the same. And we found that by varying the conditions, we could get these fibrillar structures from polylysine or polythreonine and so forth in the laboratory. And so we realized that the interactions between specific types of side chains was not important for the architecture of this, the ability to form this structure. So this amyloid structure, the fold if you like, is not encoded in the amino acid sequence. unlike the native structure. And so we began to think that this was really a different type of structure of protein molecules, that biology in general has chosen specific sequences where the side chains can interact to form particularly folded proteins, or in some cases to ensure the protein remains intrinsically disordered. But there's another form of proteins which is a polymer form, which is a characteristic of the just polypeptide chain being a polymer. And this was actually a model we worked at with Helen Sable in 1999 to show from electron microscopy, it's a model, not a structure, that you could see this array of beta sheet structures. In this case, there are 4 protofilaments that twist around each other. And we argued that the interactions between the main chain here were stabilizing this structure, as opposed to the interactions with side chains that were dominant over here. And so, although the side chains affected the exact way these structures came together, they looked essentially the same for all different types of protein. Since then, we and others have gone on to determine structures in detail. Here's one example. It's a peptide, so it has no globular structure. It's intrinsically unfolded, if you like. And by a combination of solid-state NMR, cryo-electron microscopy, and other biophysical techniques, we could determine its structure to a very high resolution, about 1 angstrom resolution. And this is the EM picture of the fibril. Actually, you can see it has an annulus of protein if you look down at the end. This is the atomic model. And again, if you look from the top, you can see there's a pair of beta strands, which then form these beta sheets that run down the length of the molecule. And indeed, you can see that more clearly if you just look at the protofilaments. And here you can see this tremendous array of hydrogen bonds that links together the peptide units into this structure. And it's this array of hydrogen bonds that gives rise to such a stable structure for this type of so-called cross-beta structure of amyloid. And now there are a number of structures of this type, and this pair of beta sheets, this double beta sheet structure, seems to be common to many of them. We wanted to analyze a lot about the structural properties of these more generally. These aggregates are not disordered in any sense. You can see how ordered they are from the structure. And actually, if you then look at the rigidity of the fibrils using, for example, atomic force microscopy, you can see that weight for weight, they're strong as steel. They're incredibly highly organized and very stable. And the main chain contributes about 75% of the structure. And in fact, I won't bore you with this, but actually if you analyze the characteristics of the rigidity, you can see again that the different proteins all fall along the same line, indicating that their essential structures are all very similar. Now, the other thing that happens when amyloid forms within people or within organs is it tends to grow. And so we looked at the force associated with that growth. And this was work by Teresa Herling with Thomas Knowles. Much of what I'll talk about is with my 2 colleagues, Thomas Knowles and Michaela Vendrusculo. And by doing microfluidics experiments, making little, if you like, pillars here, putting the amyloid structure in between, and then flowing through more aggregation-prone protein solution, these expand, and from the way they push aside these micro pillars, you can determine the force. And it's clear that the force is easily enough to disrupt cells and tissues. It's very comparable to that of many synthetic polymers. The other very surprising initially observation was that we could measure the thermodynamic stability of these fibrils. And we could do this essentially by the same techniques that are used to denature globular proteins, except you have to be very careful. And Yuji Goto was the person who really developed this method. You can increase the concentration of denaturant and watch the transition from fibrils into soluble protein and then back again. And by making these measurements on different proteins, we were able to work out the free energy of stability per residue. And one of the things that is very interesting about this is that for proteins with less than about 150 residues, the amyloid state, whether it's intrinsically disordered or folded protein, is actually more stable than that of the native protein. And so the amyloid state is more stable in these systems than is the native protein. For larger proteins, the stability per residue falls because you can get less of the protein highly organized into these beautiful, almost crystalline-like structures. So these are very stable structures. Even under physiological conditions, they can be more stable than the native protein fold. So it turns out it's not thermodynamics that matters, it's kinetics. And it's the kinetics at which aggregation occurs that is very important. And I'm going to talk about this in much more detail. But in fact, if you look at proteins in their unfolded states and measure the rates at which they aggregate, they cover well over 7 orders of magnitude. This is from as fast as you can measure to as long as you can keep a graduate student in your lab, effectively. And you can see, you can explain these. It's a polymer phenomenon on the basis of simple physicochemical parameters of charge and size and hydrophobicity. And things fall along the same line. Intrinsically disordered proteins are very reluctant to aggregate, otherwise they wouldn't be able to be present in their forms within solution. These globular proteins are much more prone to aggregate, but normally they're folded into the globular structure, and so they're protected against aggregation unless you have a mutation, for example in lysozyme, where the protein then partially unfolds, and then the aggregation propensity can be seen to be very high. So what are the biological consequences of amyloid formation? I mean, I've given you a sort of lightning tour of what we think this type of structure is about and why we think it's a very interesting form of proteins. But what consequences does it have for biology? And the first point, just to state, is that native proteins can be metastable. Again, they may not, the proteins within you may not be the most stable form of that particular polypeptide chain. And so there's an intrinsic thermodynamic propensity, particularly for small proteins and unfolded proteins and fragments of proteins, to convert from their soluble structures into these amyloid fibrils or their precursors. Fortunately, the rates of conversion are usually very slow, otherwise we'd turn into solid structures almost immediately. So you can think of it rather in this way, that when you make a protein, it's initially unfolded, and it can then do various things. It can be tangled up in the disordered aggregate, it can be degraded, it can form a partially folded state, it can form a native state, it can assemble into higher order structures. And it can also, we think in general, form these amyloid fibrils, either from fragments or intrinsically from the protein itself, particularly small proteins. To form the fibrils, it has to go through, of course, intermediate species where 2, 3, 4, 5, and then hundreds and thousands of proteins assemble into this structure. But what's very, very important to realize is that we know that biology controls all of chemistry. Now, enzymes control the chemistry of biology. They make sure you've got the right small molecule, the right metabolite in the right place at the right time to do the right thing in the right kinetics, and you end up with the right product. And proteins effectively have the same management structure, but these are a whole variety of molecular quality controls and degradation mechanisms that make sure that you have the right form of this protein at the right place at the right time to do the right function. So this is just as highly managed, we believe, as the metabolism that everyone is familiar with from organic chemistry, if you were lucky or unlucky enough to do that. But there will, and we'll hear more about this, there are some forms of amyloid which are very important functional states of proteins, but many of them, the ones I've mentioned, are associated with disease. So why are they associated with disease? First of all, they're not functional in the amyloid state in general. Secondly, they can expand to disrupt organs. But also, it turns out that particularly the small precursors of fibrils, even of proteins which have no association with disease, can form. You can see them here in electron micrographs. These are about 20 or 30 molecules in this particular type of oligomeric intermediate. And these can very rapidly penetrate cells and trigger cell death. And in fact, we've been looking at this in more detail, and you'll hear a talk from Suman Day, who's going to describe some really exciting work recently where one can detect oligomers which have toxicity by some single molecule fluorescence techniques. But essentially it means that we can look at the nature of the species which are formed and measure their toxicity in neurons or other cells. And this just shows the aggregation of A beta 42. There's a lag phase, then a rapid growth phase, and a plateau. And if you take the monomeric state, there's almost no toxicity measured here by calcium influx. If you look at the fibrils, they're relatively benign, but these species formed at the initial stages, these oligomeric structures, are extremely toxic in the sense that they very strongly disrupt lipid bilayers. And again, you can also show if you add chaperones or antibodies, you can suppress these effects dramatically. And Suman will tell you, I know a lot more about that later. Another type of experiment we've been trying to do is to understand why these types of species should disrupt bilayers. And this is some work, particularly by Giuliano Fusco, which is coming out, I think, this month in Science. And this is a very good collaboration with Alfonso de Simone, who's now at Imperial College, who has been using particularly solid-state NMR to look at the stabilized forms of various oligomers. These are from alpha-synuclein. And what one finds is that amorphous species, which are generally rather non-toxic, will often bind to the cell membrane, but they just bind to the head groups. But what turns out to be the case in these so-called amyloid oligomers that have formed, beginning to form these hydrogen-bonded internal structure and push some hydrophobic groups to the outside, is that they not only bind strongly to membranes, but they also insert the core region into the membrane, into the lipid bilayer, and so disrupt it and allow, for example, calcium influx and the other phenomena which then follow that, reactive oxygen species formation and so forth. So we've been trying to understand, again with model systems like lipid bilayers, just how these types of species could affect the nature of the cell membrane, and we've been also looking at neurons and astrocytes and so on as well. So essentially we think that all proteins are, of course, prone to aggregate. Many of them will form these quite easily, these amyloid forms, but there are a whole set of almost like housekeeping mechanisms that pick up proteins which are misfolded and pick up proteins which are aggregating, and they neutralize their potentially damaging effect. And it's the failure of these mechanisms that results in pathology, because you either get large quantities of aggregates, such as the lysozyme case, or else you get toxic species forming that induces cell death. So we're still intrigued by the fact that so many proteins in living systems, under some conditions, more than 30, 40, 50 proteins could actually form these diseases. And we began to wonder more about the nature of proteins in biology. This is a very early slide. We've now got thousands of points along this line. And what it really says is that in order to be efficient, a cell needs to pack in as many molecules as it can for its many functions and pack them close together so it can transfer information rapidly, for example, by diffusion, particularly by diffusion. And so if you need a protein at high concentration, it has to be soluble. And so if you only need it at low concentration, it has to be less soluble. And so we found a linear correlation between the log of the rate of aggregation and the log of the expression level, where all the proteins that we could see at that time fell along this line. The reason is it's very hard to make proteins soluble. You have to put the right residues at the right place to design the surface very carefully. Random mutations tend to make the protein stickier and aggregate. And so you end up sitting right on this edge of, if you like, the solubility between the safety region here and the disease region here. And this is work, like much of the I'll talk about now, work I'll talk about with Michele Vondrusculo. It also turned out that we've started scanning the human genome and doing proteomics and transcriptomics to look about the solubility, the predicted solubility which we can generate from these algorithms relative to the expression levels. And what we found is, and this is work with Rick Morimoto at Northwestern as well, that there are a number of proteins which actually are present at much higher concentration than that line would suggest. And when we've looked where they are, they tend to be in these regions, in these pathways associated with neurodegenerative disorders. And so here's a pathway for Alzheimer's disease, and it's not just A beta which becomes aggregation prone, it's actually the proteins in the mitochondria, in other aspects of the pathway, which are, again, really supersaturated, very metastable. So when one protein starts to aggregate and the control is lost and homeostasis is lost, then they all tend to aggregate. And so we are right on the edge of this aggregation mechanism, and these housekeeping mechanisms do their best, but they don't always function. And why don't they function? Well, these diseases are relatively modern diseases, and it's because we live much longer than we used to do. You know, the average lifespan is now much greater than it was, and you know, the risk of Alzheimer's disease goes up dramatically over the age of 65, I'm sorry to say. But at the age of 85, there's almost a 1 in 2 chance of getting this disease. And of all the people in the history of the world who've reached the age of 65, it's estimated that most of them are alive today. So this is a huge change in demography. In Britain, we thought it was a good idea to feed young cows and old cows, and generated a prion disease because the aggregates are already present. And the dietary changes for so many people generating obesity generates, again, changes in levels of hormones and other proteins. And even putting people on dialysis changes the levels of proteins. So we thought these diseases must be linked to very subtle changes in these aggregation propensities, linked to failures of quality control, perhaps associated mutations that make them much more aggregation prone. And then you can get this widespread loss of protein homeostasis. And so to test this, one of the things that we tried to do is to modify the aggregation propensity. And this is for A beta 42. Through the work of Fabrizio Chiti, we knew we could make single point mutations and change very significantly the propensity to aggregate. So we could put in, say, a charge group and it reduced the propensity to aggregate, and we could put in a hydrophobic group and it would increase the propensity to aggregate. And then we put these into Drosophila so that you could see the effects. It's a sort of Drosophila model of Alzheimer's disease. And so you can see, actually, that the wild-type protein at this length of time, the deposits are forming, causing neuronal damage, and the flies are struggling. You bang these test tubes on the bench and watch the flies climb up again. Here we increase the aggregation propensity by 1 or 2%. The flies are not functional, but if you reduce it by a few percent, then they're perfectly normal. And so you get this wonderful graph straight away from these results. But what's particularly exciting is this is physics and this is biology, and there's a linear relationship between the two. A change of just a few percent in a physical property can overwhelm the 20,000 genes in the organism which are protecting us against it. So the last part of my talk, I want to talk about the mechanism of amyloid formation and how it relates to potential therapies and disease. And we now know that amyloid formation involves a series of highly coupled events, and this is work particularly from Thomas Knowles' group. So you have to start off with the soluble form as a primary nucleation step. There's then, for example, elongation and sometimes dissociation. And then there's secondary events. If a fibrillar structure fragments, then you double the number of ends and it will grow twice as quickly. And also, the fibrils can be sticky and they attract monomers, and this then nucleates on the surface. So we know that there are lots of these function. We thought we'd try and look at them. And I just want to give one example of this, which is to try and watch this initial event in aggregation. This is notoriously difficult to follow, the primary nucleation, but we realized that if you made the volume small enough, you might be able to make the nucleation events rare enough to measure. And so this is a microfluidics experiment. You form very small droplets of aqueous solutions surrounded by oil. We put them into a storage array which extends them into sort of columns. And then if you look at the results, you could say these are different droplets. And primary nucleation is a stochastic process. And you can see they've got a dye in here, and when the amyloid nucleation forms, they light up one by one. And we're watching the primary nucleation event. More than that, if you look at a single droplet, you can see that when you get a nucleation event, for example here, it will spread because there'll be diffusion of the aggregates and then they'll nucleate elsewhere. So we could follow these processes in principle, but we really wanted to analyze the multiplicity of processes that follow this. How do we look at the growth and the fragmentation and so forth, and even the spreading of this process? And this was a real breakthrough by Thomas Knowles when he was a graduate student with myself and Mark Welland in the Nanoscience Center, because he looked at the different steps in this process, and they're all a series of coupled differential equations, nonlinear differential equations. They were thought to be insoluble. They were stated in every review to be insoluble, but Thomas solved them. And by taking, for example, data on the aggregation kinetics, this just shows a protein, low concentration, you increase the concentration, the aggregation is faster. And if you can fit the experimental data, you can get information on these individual rate constants. So suddenly, we had the ability to look at these microscopic steps. And here's one example of this. This is A beta 42. You don't need to see any of the details, fortunately, on this. It's just, this is work with Sarah Linzer and Lund. We were able to do a concentration dependence of the aggregation. We also varied the conditions, for example, putting agitation on. But if you just look at this set of data under quiescent conditions, as you change the concentration, the aggregation kinetics change, and you can then fit them to this analysis to generate the kinetic parameters. And it turns out that to our surprise, it's not primary nucleation which is the problem for A beta 42, it's actually secondary nucleation. So when you get aggregates, they catalyze the formation of more. That's why you get this explosive exponential growth in aggregates, because each aggregate that forms then catalyzes more aggregates. So in the last few minutes, which I think Priyanka will let me have, I've got 10 minutes according to my timer, which may be different from yours. The question is, if all these things are happening, why do we manage to live as long as we do without these processes happening? What is it that protects us against these processes, which in the laboratory we can see in hours or days or so on? And I just want to give 3 examples of things that biology has actually, we think, has done to make this it possible for us to avoid these diseases for so long. The first is that if you reduce the volume of a compartment, then you reduce the probability of nucleation. You only need one nucleus forming. So if it's small, the probability is much less than the whole of this room. So biology compartmentalizes its proteins, and that hugely increases the kinetic stability, either in cells or in separate compartments. Another thing that we've done, I didn't have time to talk about it, is that with Rick Morimoto, Michaela Vendriscolo's work, particularly Prajwal Sirium, we found that if you compare tissue from people with disease and without disease, that actually the most aggregation-prone proteins, these metastable ones, are actually down-regulated to maintain proteostasis. So if you look at the most dangerous proteins against down-regulation, Parkinson's, Huntington's, Alzheimer's, all up at the top, along actually with oxidative phosphorylation involving the mitochondria, which is a central process. And so biology has a protective mechanism for reducing the concentration of proteins that are aggregation prone if necessary. And the other thing it does is exploit molecular chaperones, which are, as you know, important in protein folding. And these can suppress the aggregation of proteins at much later stages. And so this is A-beta 42. This is a chaperone called Bricos. It reduces the rate of aggregation when you add it to A-beta 42. And because we can analyze the kinetics, we can tell exactly how it happens. It happens because this chaperone inhibits this crucial secondary nucleation step, the surface-induced aggregation, and then dramatically reduces the number of oligomers that you get. And it doesn't, you can see from this, it doesn't affect primary nucleation or elongation, just this particular step. We know how it does it. It binds to the fibrils and coats them, and therefore inhibits the aggregation. And this binding then massively suppresses toxicity in these experiments in mouse brain tissue. So this is without the chaperone, and with the chaperone, it takes it back virtually to a non-toxic species. And it turns out that other chaperones can inhibit other steps in the mechanism of aggregation. And we know that of the hundreds of chaperones we have, some inhibit primary nucleation, some inhibit even elongation, some may inhibit fragmentation, and certainly, as I've shown, some eliminate this or inhibit strongly this secondary nucleation, which is so important for the proliferation of disease-associated systems. So in the very last section of the talk, it's can we mimic these effects? Could we design molecules that would do the same sorts of thing that chaperones do, because they would then be potential therapeutics? And here's one beautiful example from Michaela Vendriscolo's lab, where, by Francisco Aprile particularly, he's taken the A-beta 42 system and he's designed a single chain antibodies that bind specifically to different parts of the chain. And this is done in a computer. I can tell you more later if you want to know how it's done. But we could show experimentally these particular antibodies, the 5 shown here, will bind to different sections of the A-beta 42. And what's remarkable then is if you look at the effects of these designed antibodies on the kinetics of these microscopic steps, they have very different effects. So those which bind to the N-terminus inhibit primary nucleation dramatically. There are others which inhibit secondary nucleation. So depending on where they bind to the molecule and what state of the molecule they bind to, these different antibodies can change dramatically the kinetics of different steps. Of course, we'd like to look at these not just in the test tube, but in vivo, and the model organism. I think Johnny Habchi may talk about this. We've used C. elegans. We've got a high throughput method of studying their behavior. And in fact, I'll just talk to this one really. If you look at the effects in vivo, there's a very similar effects that you see in vitro. For example, the antibodies which inhibit the primary nucleation, you have to apply very early in the lifespan of the C. elegans, while those which inhibit the secondary nucleation, you can apply even later in the aggregation, because once primary nucleation has occurred, then if you can't stop anything later, then it will just continue to aggregate. But secondary nucleation, if you stop it later, it will stop the proliferation. And we've gone on to look at small molecules, and Johnny Habchi and Priyanka Yoshi will give talks about this work, so I'm not going to say anything really very much about it, except we found small molecules which have huge effects on the aggregation kinetics. And they can act in similar ways to the chaperones and antibodies by inhibiting these individual steps, either by inhibiting, for example, primary nucleation or binding to the surface and inhibiting secondary nucleation. And so we can generate a screening strategy I think, again, Priyanka will talk about this. We get a library of molecules, either synthetic or in her case, natural products. We look to see their effects on aggregation. If they inhibit aggregation, we look to see which step, and then we put them into C. elegans initially and see what they do. And this just gives you one example. These are A-beta transgenic Drosophila C. elegans. They're very unhappy. They're almost paralyzed. But if you put, this is actually, it's a approved cancer drug at low concentration into the solution, then it prevents this process happening. And we think it happens by preventing the primary nucleation step. You have to give it right at the beginning of the process. You know, it's fantastic news for demented worms. We know we can cure it completely. And we hope we might be able to do the same with humans. So to finish, we think there are many, many opportunities to intervene in this process. If we could stop primary nucleation very effectively, we really wouldn't have to worry about anything else. But then all these other steps, rather like in biology, if you can't stop primary nucleation, you can try and stop the proliferation of the aggregates. And if we can find a range of these species, then they could actually be potential drug leads. As I said, these are really generic approaches. This is, again, C. elegans transgenic for alpha-synuclein associated with Parkinson's disease. This is a natural product that came from Michael Zaslav's group in Georgetown. And if you give that to the solution, you can again suppress completely all the paralytic effects of the alpha-synuclein, and you get no aggregates forming here. And we know why this happens. It displaces alpha-synuclein from the membrane surfaces. So we've been very lucky. We've raised money. We're now setting up this center, and actually this building is almost complete. And the top 2 floors will be for basic science, and the ground floor will be for a drug discovery company that is already established, and Johnny Habchi is very much involved with that. And so we're hoping we can take these sort of basic science into compounds, which then we will take into animal models, and then, with luck, into humans. And so the real message of what I've talked about is that we really tried to look at these fundamental principles, taken lots of techniques, tried to work out how misfolding aggregation occur, how normal biology inhibits this, and whether we can use this then to develop a series of novel therapeutic strategies for these diseases, which are so prevalent. And this just shows that we started by accident. This shows that in the US, for example, the US will spend $20 trillion, which is more than 10 times their annual exports, just caring for people with Alzheimer's disease in the next generation, if we can't cause this to come down by finding some therapy. Indeed, 70% of the cases of Alzheimer's in the next 35 years will be in developing and relatively low-income countries, middle-income countries. So I sometimes say I did virtually everything myself, but a few other people helped. So we have a fantastic set of international students. And what's been really important for us is we have a lot of collaborators all over the world who have been involved in all the experiments I've talked about, from Martin Karplus as a theoretician through to David Lomas as a clinician. And particularly Thomas Knowles, Michaela Vendruscolo in Cambridge, Sarah Linzer in Lund. We work very closely together. And I wanted to acknowledge Fabrizio Chiti in Florence. We still work with him, and he really was one of the people who got us involved in this area to begin with. So I probably talked at least long enough. So I hope that I gave you a picture of this area. I hope that many of the ideas we picked up in much more detail later. And thank you very, very much for the invitation and for your attention.
We can take a couple of questions.
possible to use a protein as a bait, and since the aggregation will not be dependent on side chains but on the main chain, capture all proteins that exist in any condition inside an organelle or in a cell or in the serum or elsewhere, and then do proteomics to identify all the proteins that are seen associating with an aggregate that is primarily constituted of one single protein that's overexpressed and also prone to aggregation, so that we could finish identifying all the proteins in the human proteome that are likely to go and bind to amyloids which are forming because of some other protein.
If I answer the second part first, we do hope that it may be possible, particularly for these later stages, to find molecules which act on a range of different aggregation-prone proteins. So they will suppress many different stages. Of course, that's what chaperones do. They affect many different proteins. In terms of finding a protein that captures others, apart from perhaps poly-Q proteins, I think it's very unlikely. And the reason is that the side chains don't affect the basic architecture, but they affect the spacing of the sheets and the exact way that the fibrils assemble. And these are very light crystals, and it's a bit the same question. If you mix two proteins together, do you get a mixed crystal? And the answer is you generally get phase separation, the crystals of each of them, because they're more highly organized in this structure. So I think it's unlikely to find a bait in that sense, but actually I think there are other ways in which you can potentially find, if you like, really generic systems that will affect a large range of aggregation-prone proteins.
Here. So you, Professor Dobson.
As I understand, your screening strategy involves sort of in vitro screening for inhibition of aggregation and then going into the worms for the in vivo analysis. Have you considered sort of phenotypic screening with that system to possibly identify compounds that modulate the homeostasis machinery?
Absolutely. We're not just in worms, but also in individual cells. I mean, ideally, what we'd like to do is to find molecules that return the homeostasis back to its perfect level. And we have the potential to do that now. Absolutely, absolutely. No, it's a very, very good question, and I'm sure it's an incredibly important strategy which we're just beginning to look at.
Yeah, Professor Dobson. So your talk mostly focused on the fibril formation, and you have
speak a little louder, sorry.
Sorry?
Could you speak a little bit louder?
Yeah. Your talk mostly focused on fibril formation, and you have some fantastic strategies to take care of those. But there is some evidence that it is the oligomers and not the fibrils that could be the most toxic species. So in some cases, stopping the fibril formation may not be wise. Could you care to comment on that?
Absolutely, and I probably was not very clear on that, because what we're actually trying to do is to reduce the number of oligomers. But it turns out, in A beta, for example, once you've got the fibrils formed, they catalyze the formation of oligomers. It's the oligomers which are clearly the unpleasant species that interact inappropriately. They have very sticky surfaces, a high surface to volume. And so I didn't intend to imply that we were trying to stop the fibrils forming. We're trying to stop them catalyzing other aggregates. And we're also trying to stop, for example, a primary nucleation, the oligomers forming in the first place. And so fibrils are less harmful than others. It's the oligomeric species that we're trying to suppress. And indeed, that's why I focused on the nucleation steps, the primary and secondary nucleation, because those are the steps which form nuclei which grow into oligomers.
Thank you, Professor Dobson.